Welcome back to the deep dive. Today, we're embarking on a fascinating journey into the microscopic world of some of the body's most vital internal organs. Think of them like incredibly complex, bustling factories working tirelessly behind the scenes.
Indeed. We're taking a closer look at the liver, gallbladder, and pancreas organs, often discussed together, because their roles in digestion and metabolism are so tightly linked.
Yeah, they really work as a team.
They do. Our mission today is to really unpack the key insights from their microscopic anatomy, explore their specialized cells, and see how their structure directly impacts their incredible functions. And we'll definitely touch on the clinical correlations, too.
So if you've ever wondered how these internal powerhouses really work at the cellular level, or how they handle everything from nutrients to toxins, or maybe what happens when things go wrong, then this deep dive is definitely for you. Let's get into it. We're starting our tour with the liver, as the undisputed heavyweight champion of your internal organs.
Absolutely. Largest glandular tissue in the body, weighs about 1,500 grams. It's mostly in your upper right abdomen, kind of tucked safely behind the ribs.
Right.
And it's wrapped in something called Glisson's capsule. That's a tough, fibrous layer. And then there's a slippery outer coat, the visceral peritoneum. Now, anatomically, we talk about lobes, but functionally,
More about the blood supply and bile drainage, isn't it?
exactly. That's
Yeah.
what really matters clinically. How the blood gets in and the bile gets out dictates how we think about its sections in practice.
And it's amazing how it all starts,
Yeah.
way back in the embryo. Just a tiny little outpouching from the early gut, the duodenum.
Yeah, the hepatic diverticulum. And from that little bud comes the main liver cells, the hepatocytes, and eventually that whole complex network, the biliary tree. Pretty incredible development.
So on to its jobs. You called it a multitasker
Oh,
earlier.
it really is. A chemical factory running 24/7. It makes most of your circulating plasma proteins, think albumin, crucial for keeping fluid in your blood vessels. And also lipoproteins like VLDLs, LDLs, HDLs. These are basically the delivery trucks for fats and cholesterol around your body.
It also makes glycoproteins needed for things like iron transferrin, right?
Mhm.
And even blood clotting factors, prothrombin, fibrinogen,
Essential stuff. Without the liver making those, clotting would be a huge problem.
and vitamins.
Yep, it's a vital storage and conversion hub for the fat-soluble ones, A, D, E, and K. Vitamin A for vision, D for calcium, E as an antioxidant, K for those clotting factors we just mentioned. Deficiencies here cause real issues.
Like night blindness or bleeding problems.
Exactly. And it's key for metals, too, especially iron and copper. Hepatocytes are the main long-term storage depot for iron.
So too much iron, say from lots of transfusions, can overload the system, leading to things like hemochromatosis.
Precisely. The storage capacity isn't infinite, and overload damages the liver. And copper metabolism is just as tightly regulated.
You mentioned Wilson disease earlier.
Yes, a mutation in a single gene, ATP7B, messes up copper transport. Copper builds up in the liver, the brain. It's devastating. It just highlights how incredibly precise these metabolic balances are.
Okay, and detoxification. That's a big one everyone associates with the liver.
It is. It's our primary detox center. It takes drugs, toxins, and converts them into forms that are easier to get rid of, usually more water-soluble.
So the kidneys can flush them out.
Right. Often uses a two-phase process involving enzymes like the cytochrome P450 family. These are mainly in the smooth ER and mitochondria of the liver cells.
So it's chemically modifying these substances.
Exactly. And metabolically, the liver is central. It manages blood sugar through glycogen storage and release. It handles fatty acids, makes ketone bodies, deals with cholesterol. It metabolizes amino acids, makes urea. It really orchestrates the whole metabolic symphony.
Wow, and it makes bile too, right? That's
Yeah.
its exocrine job.
Correct. Bile production. That greenish yellow fluid needed for fat digestion and getting rid of waste like bilirubin. It flows out through that complex biliary tree we mentioned towards the gallbladder and intestine.
Does it have any hormone-like roles too?
It does, sort of endocrine-like functions.
Yeah.
It modifies hormones made elsewhere, like converting thyroid hormone, thyroxine, into the more active form, T3.
Okay.
And it produces IGF-1, insulin-like growth factor 1, which amplifies growth hormones effects. This is actually really critical. There are genetic IGF-1 deficiencies.
Like in the Bayaka pygmies.
Yes, exactly. They have normal growth hormone levels, but without enough IGF-1 from the liver, their pubertal growth spurt is significantly reduced, leading to short stature. Shows how the liver links into the endocrine system.
Okay, so for an organ doing so much, Its blood supply must be pretty special. You mentioned a dual supply.
It's unique, yeah. About 75% of the blood flowing into the liver comes via the hepatic portal vein. This is venous blood coming straight from the intestines, pancreas, spleen.
So low on oxygen, but packed with nutrients
Hmm.
and maybe toxins, just absorbed from lunch.
Precisely. It's bringing everything the gut just absorbed directly to the liver for processing first. The other 25% of the inflow is oxygenated blood from the hepatic artery.
And these two mix.
They do.
Yeah.
They mix before they perfuse the actual liver cells, the hepatocytes. So liver cells never see fully oxygenated arterial blood by itself. They work in this mixed environment.
Interesting. And these vessels travel together.
Often, yes. Along with bile ducts and lymphatic vessels, they form what we call a portal triad, usually found at the corners of the liver structural units. It's a key landmark on a histology slide.
So let's zoom in. If we looked at a liver slide, what's the basic structure?
You'd see the parenchyma, that's the functional tissue organized into plates of hepatocytes, usually just one cell thick in adults. And these plates are separated by tiny blood channels called sinusoids.
Okay. And you mentioned models to understand the organization.
Right, three main ways to think about it conceptually. First, the classic lobule. It's the easiest to spot, looks kind of like a hexagon.
With a central vein right in the middle, and those portal triads we just talked about at the 6 corners, hepatocytes radiate outwards from that central vein.
Like spokes on a wheel.
Kind of, yeah. Then there's the portal lobule. It's more triangular, and it emphasizes bile flow, with the bile duct of a portal triad at its center.
But the third one, the sinus, is the most important clinically.
Arguably, yes.
Yeah.
The liver sinus, it's more diamond or lozenge shaped. And it gives the best correlation between blood flow, metabolic activity, and crucially, where damage occurs in disease.
Because of the zones.
Exactly. The sinus is divided into 3 zones based on how close the hepatocytes are to the incoming blood supply from that portal triad. Zone 1 gets the freshest, most oxygenated, nutrient-rich blood.
And zone 3?
Zone 3 is furthest away, near the central vein, so it gets blood that's already had oxygen and nutrients pulled out. It receives the least oxygenated blood.
And this zonation matters hugely in disease.
Absolutely critical. For example, think about congestive heart failure. Blood flow is sluggish. Oxygen delivery is poor. Which zone suffers first? Zone 3, because it's already getting the least oxygen.
Precisely. You see ischemic necrosis, cell death due to lack of oxygen, specifically in zone 3. We call it centrilobular necrosis.
And acetaminophen overdose. You mentioned that targets zone 3 too.
Yes, the toxic metabolite produced from acetaminophen primarily damages zone 3 hepatocytes. It can cause massive cell death there, leading to acute liver failure. So understanding these zones is, well, it's vital for diagnosis and predicting outcomes.
Okay, let's dive deeper into those sinusoids and the cells around them. You said the sinusoid lining is discontinuous.
Yeah, the endothelial cells lining the sinusoids are thin and have large gaps, fenestrae, between and even through them. It's not a tight barrier like in most capillaries. This allows really easy exchange of molecules between the blood and the liver cells.
And inside these sinusoids are the Kupffer cells.
Right. Kupffer cells, they're specialized macrophages, part of the mononuclear phagocyte system. They actually sit right in the sinusoid lining, sampling the blood flowing past.
Like little Pac-Man gobbling up debris.
That's a good way to think of it. They remove old red blood cells, bacteria, cellular debris, and they recycle iron. Very important cleanup crew.
And between the sinusoid lining and the hepatocytes themselves, there's a space.
Yes, the parasinusoidal space, or a space of Dis. A very narrow gap, but incredibly important. This is the primary site where exchange happens. Blood plasma filters into this space.
And the hepatocytes interact with it there.
Exactly. Hepatocytes have tiny, irregular microvilli sticking out into the space of Dis, massively increasing their surface area for absorption and secretion.
And there's another cell type hiding in there, the stellate cells.
Yes, tucked away in the space of Dis are the hepatic stellate cells, also called Edo cells. Normally, in a healthy liver, their main job is storing vitamin A. They're pretty inconspicuous.
So they change dramatically in disease.
They undergo a remarkable transformation. In response to chronic liver injury, like from chronic hepatitis, alcohol abuse, fatty liver disease, these quiet vitamin A storing cells get activated.
Activated how?
They turn into myofibroblasts, cells that are very active in producing collagen and other extracellular matrix components.
So they start laying down scar tissue.
Precisely. They become the main drivers of liver fibrosis, that scarring process that eventually leads to cirrhosis if the injury persists. It's fascinating and devastating how this one cell type can switch roles and contribute so heavily to liver failure. It really makes you think, how can such a small player have such a huge impact?
A crucial question. So the hepatocytes themselves, what are they like up close?
They're large cells, often polygonal, making up about 80% of the liver mass. You often see two nuclei in one cell, reflecting their high metabolic activity. Their cytoplasm is just packed with organelles.
Like what?
Tons of mitochondria for energy, extensive rough ER for making proteins like albumin, lots of smooth ER.
For the detoxification and cholesterol synthesis.
Exactly. And the smooth ER can actually grow hypertrophy if it's constantly dealing with substances like alcohol or certain drugs. The cell adapts, tries to cope. They also have a very prominent Golgi apparatus for processing and packaging molecules.
You also mentioned peroxisomes.
Yes, loads of them, maybe 200 to 300 per cell. They're involved in breaking down fatty acids, but also play a key role in detoxifying alcohol. They handle about half the ethanol you might ingest, converting it to acetaldehyde. So detoxification isn't just the smooth ER's job.
Okay, now let's trace the path of bile after it's made through the biliary tree.
Right. It's an intricate network, over 2 kilometers if you stretched it all out. Starts tiny. The smallest branches are the bile canaliculi. These aren't true ducts. They're just tiny grooves formed between adjacent hepatocytes.
Sealed off.
Yep, by tight junctions so bile doesn't leak out. And they have microvilli suggesting active secretion into the canaliculus.
Where do they drain?
Into short little channels called the canals of Herring. And these are really interesting structures.
Why is that?
Because they're lined partly by hepatocytes and partly by the first cells of the duct system proper, called cholangiocytes. And more importantly, this junctional area, the canals of Herring, is thought to be a niche for liver progenitor cell stem cells.
Liver stem cells. Wow. So potential for regeneration.
That's the idea. Their location is perfect, right at the boundary between the liver cell plates and the bile ducts. It raises a huge question. Could we harness these cells therapeutically for liver repair? It's a major area of research.
Definitely something to watch. So from the canals of Herring.
Bile flows into intrahepatic bile ductules, then into larger interlobular ducts. Those are part of the portal triads, which eventually merge to form the main right and left hepatic ducts that carry bile out of the liver.
And these ducts are lined by.
Cholangiocytes, specialized epithelial cells. And they aren't just passive pipes. They modify the bile, absorb or secrete water and electrolytes. They even have a primary cilium, like a little antenna, that senses bile flow and composition, and they adjust their function accordingly. Quite dynamic cells.
Okay, so bile leaves the liver via the hepatic ducts. Where does it go next?
Most of it heads to the gallbladder via the cystic duct. The gallbladder is that small, pear-shaped sac nestled underneath the liver, holds about 50 milliliters.
And its job is storage and concentration.
Exactly. Its main function is to concentrate the bile, primarily by removing water up to 90% of it. This makes the bile much more potent for when you need it to digest fats.
And how does it get released?
When fat enters your duodenum, the first part of the small intestine, it triggers the release of hormones like cholecystokinin, CCK. CCK signals the gallbladder's smooth muscle wall to contract forcefully.
Squirting the concentrated bile out.
Precisely. It goes down the cystic duct, into the common bile duct, and then into the duodenum to mix with food.
What does the gallbladder lining look like microscopically?
Its mucosa, the inner lining, is thrown into numerous deep folds which disappear when it's full and distended. It's lined by simple columnar epithelial cells, cholangiocytes again, specialized for water absorption.
How do they absorb water so well?
They're packed with mitochondria to provide energy for active transport, and they have complex folds on their sides, lateral plications, plus special water channels called aquaporins, all designed for rapid water movement out of the bile and into the tissue.
Interesting. And those Rokitansky-Aschoff sinuses you mentioned.
Ah, yes. These are deep little outpouchings, invaginations of the mucosa that can push down into the muscle layer. They're considered a normal variation, but, well, they can sometimes harbor bacteria or contribute to chronic inflammation, and they might increase the risk of gallstone formation in some people. It's one of those things where a seemingly minor anatomical feature might potentially set the stage for problems later on. Makes you wonder about other subtle variations, doesn't it?
It does. Okay, so we've covered the liver and gallbladder. Let's move to the pancreas, the third player in this trio, Dual role, you said.
Absolutely. The pancreas is an elongated gland, kind of sits in the curve of the duodenum. It's really two glands in one. It has a major exocrine component.
Making digestive juices.
Exactly. Powerful digestive enzymes secreted into the duodenum to break down food. And then it has a crucial endocrine component, the islets of Langerhans.
Making hormones like insulin.
Right. Insulin, glucagon, and others secreted directly into the blood to control metabolism, especially glucose levels.
Let's look at the exocrine part first. What's its structure?
It's made up of secretory units called acini. These are clusters of pyramid-shaped serous cells. Under the microscope, their apical cytoplasm, the part facing the central lumen, is packed with bright pink staining granules.
Zymogen granule.
Yep, zymogen granules. These contain the inactive precursors of the digestive enzymes, proteases for protein, amylase for carbs, lipase for fats, nucleases for DNRNA.
And they're inactive inside the pancreas.
Critically important, they only become active once they reach the small intestine. There's an enzyme in the duodenum, enterokinase, that kicks off the activation cascade. This prevents the pancreas from digesting itself, which would be, well, catastrophic pancreatitis.
A clever safety mechanism. Is there anything unique about the ducts starting from these acinas?
There is. The very first part of the duct system, the intercalated duct, actually starts inside the acinus. The cells forming this initial segment right in the center of the acinus are called centroacinar cells.
Okay.
They stain quite pale and are a key feature to look for when identifying pancreatic tissue. They're unique to the pancreas.
And what do these ducts do besides carry the enzymes?
The duct cells, starting with the centroacinar cells, and especially the intercalated ducts, secrete a large volume of fluid that's rich in bicarbonate.
Bicarbonate to neutralize stomach acid.
Exactly. The chyme coming from the stomach is highly acidic. This bicarbonate neutralizes it, creating the slightly alkaline pH that the pancreatic enzymes need to work effectively. Absolutely essential for digestion.
Okay, now for the endocrine part, the islets of Langerhans.
Right. Scattered throughout the exocrine tissue, you find these little islets of paler staining cells. These are the endocrine factories. They're richly supplied with fenestrated capillaries.
So hormones can get into the blood quickly.
Precisely. Quick release is key. And within these islets, there are several cell types. The main players are beta cells, making up maybe 60–70%. They secrete insulin.
Insulin lowers blood sugar.
Correct. It signals cells, especially liver, muscle, and fat, to take up glucose from the blood. Then you have alpha cells, maybe 15–20%. They secrete glucagon,
which raises blood sugar.
Yes, mainly by telling the liver to release stored glucose and make new glucose. And then there are delta cells, 5–10%, secreting somatostatin.
What does that do?
It acts locally within the islet to inhibit the release of both insulin and glucagon, kind of a local regulator or brake. There are other minor cell types too, but those are the big three.
It's amazing that these tiny clusters, just 1–2% of the pancreas, control glucose for the whole body.
It really is. Their hormones are incredibly potent. Take insulin. Its synthesis is quite a process. Starts as pre-proinsulin, gets processed to proinsulin, and then the final step cleaves off a fragment called the C-peptide, leaving active insulin.
And measuring that C-peptide is useful clinically.
Very useful, because C-peptide is released in equal amounts to insulin produced by the pancreas. Measuring C-peptide levels in the blood tells doctors how much insulin your own pancreas is actually making. This is really helpful in managing diabetes, distinguishing type 1 from type 2, for instance.
So it's a marker of your B-cell function.
Exactly. And the regulation of these islet cells is complex, too. Blood glucose is the main driver, of course, but other hormones and even the nervous system play a role.
Like fight-or-flight response affecting insulin.
Right. Sympathetic stimulation tends to inhibit insulin release. You want glucose available during stress. Parasympathetic stimulation tends to increase both insulin and glucagon. It's a finely tuned system.
And you mentioned a connection to Alzheimer's.
Yes, it's an area of ongoing research, but there's growing evidence suggesting that problems with insulin signaling in the brain, reduced expression of insulin or its receptors, might contribute to the neural degeneration seen in Alzheimer's disease.
Wow, a link between pancreatic hormones and brain health.
It's quite profound. It implies that metabolic health and cognitive health might be much more interconnected than we previously thought. It really opens up questions, doesn't it? What new therapeutic avenues might this connection reveal for neurological diseases?
Absolutely fascinating. Wow, what a deep dive that was into the liver, gallbladder, and pancreas. We've really peeled back the layers, haven't we, to see the incredible microscopic details that allow them to perform their vital jobs.
We really have, and hopefully it's clear how understanding the individual cells, the hepatocytes, the cholangiocytes, the Kupffer cells, the islet cells, and how they're organized, like those liver zones or the pancreatic Ashini, is absolutely crucial. It helps us grasp their physiology and what happens in disease.
It's just remarkable how structure and function are so tightly interwoven at this level. Makes you appreciate the sheer complexity and, well, the resilience of the body.
It does. This histological journey reminds us that these tiny components matter immensely for our overall health. And the constant communication hormones, nerves, local signals within and between these organs highlights how adaptive they are, but also how vulnerable they can be when things get overwhelmed.
So, what's the key takeaway for you listening? Well, next time you think about your diet or maybe a medication you're taking, just take a second to appreciate the incredible unseen work happening inside you. Your liver, gallbladder, pancreas, they're running complex microscopic factories 24/7 to keep you going.
And maybe consider this thought. We saw how the humble hepatic stellate cell can completely change its identity under stress and drive liver scarring. It makes you wonder what other seemingly quiet cells in our bodies might hold similar undiscovered power to influence our health or disease, just waiting for us to understand their secrets.
A really provocative thought to end on. We hope this deep dive has given you a new appreciation for these often unsung heroes of your digestive and metabolic systems. Thanks for joining us. Until next time.